This presentation is on the toxin nicotine via the respiratory system as the route of entry and targeting the hepatocytes in particular. Nicotine is a tertiary amine consisting of a pyridine and a pyridine ring. Its chemical name is S3,1-methyl-2-pyridinol pyridine and has a chemical formula of C10H14N2. The toxin itself is colorless to pale yellow and turns brown on exposure to air or light. Nicotine is water-soluble and strongly alkaline. Nicotine has a very distinct odor that is commonly recognized. Nicotine exists as two isomers, with S-nicotine being the more active form, as well as a prevalent form in tobacco and in Nicotrol inhalers. The toxin is naturally occurring and is obtained from harvesting tobacco plants. Nicotine represents 5% of a tobacco plant. Nicotine can come into contact with the body through the GI tract, the respiratory system, and dermally. In this presentation, the focus is on the respiratory system. Typically, nicotine is admitted into the respiratory system via either smoking or breathing in secondhand smoke. The absorption of nicotine is directly related to its physical-chemical properties. As can be seen from its structure, nicotine is an organic molecule consisting of a pyridine molecule and a cyclic ring including a tertiary amine. There are thus two ionizable groups, the groups comprised of the nitrogen atoms, with pKa values of 7.84 and 3.04. Based on the known pKa values of tertiary amines and pyridine, it can be determined that the pyridine will have the lower pKa value of 3.04 and the tertiary amine will have the higher pKa value of 7.84. The tertiary amine will thus be protonated and cationic at physiological pH 7. The pyridine will be deprotonated and will remain neutral. Overall, the molecule will be cationic. The log POW coefficient shows that nicotine is 94% soluble in octanol and 6% soluble in water. This ratio is within the drug-like range, therefore it is bioavailable but is a hydrophobic molecule. In terms of polarity, amine groups are ranked 6th on the polarity scale. Therefore, the molecule is not very polar. This lack of polarity of the molecule facilitates absorption of nicotine. Overall, transcellular diffusion is preferable for neutral nicotine, and the cationic nicotine species prefers paracellular diffusion. Nicotine is rapidly absorbed in the lung alveolar epithelium because the blood flow is high in the lung capillaries. Once nicotine is absorbed, it is easily and rapidly distributed throughout the body. Initially, nicotine is inhaled and is small enough to bypass the cilia and mucus barriers. It proceeds to the trachea, to the right and left bronchi, which then branch into the bronchioles to a cluster of alveoli. Gas exchange occurs in the alveoli, where blood receives oxygen from the alveoli. The red blood cells then travel through the pulmonary capillaries where they give up CO2 and pick up O2. The oxygenated blood then travels to the pulmonary veins to the heart via a one-way valve into the left ventricle. The toxin then leaves the heart through the aorta, and the toxin continues to travel through its major branch, the celiac artery. This artery leads to the hepatic artery, which goes to the hepatocytes, which is the ultimate target. Nicotine is mainly metabolized in the liver, and hepatocytes are the main cells of the liver. The primary metabolites of nicotine are cotinine and nicotine N-oxide, which are products of the hepatic oxidation of nicotine via cytochrome P450s. Specifically, cytochrome P2A6 is the major hepatic P450 enzyme that catalyzes the pathway of nicotine metabolism. This metabolism consists of C-oxidation to cotinine. Oxidation, which is phase 1 metabolism, causes an increase in hydrophilicity, which facilitates excretion and helps to remove the toxin from distribution. Both nicotine and its metabolites are rapidly eliminated by the kidneys. The rate of urinary excretion of nicotine depends on the pH of the urine, and excretion decreases when the urine is alkaline. Nicotine may also be excreted in milk of lactating women. Approximately 10 to 20% of the absorbed dose of the drug is excreted unchanged. The excretion of unchanged nicotine is not metabolism dependent. Nicotine can also be excreted through the urine, which is metabolism dependent as nicotine is produced through the oxidation of nicotine via cytochrome P450 enzymes. Nicotine is thus excretable, but unfortunately is able to be reabsorbed. The parent toxin is nicotine. The effector toxins are nicotine and a nitrosamino type molecule. The parent toxin, nicotine, in itself is very toxic. The nitrosamino type molecule that results from the pearl ring opening is also toxic. The target of the toxin is the hepatocytes. The hepatocytes are metabolically active liver cells, and the liver is the organ responsible for helping to maintain homeostasis. Hepatocytes are very important for bioactivation and detoxification. The hepatocytes consist of a large amount of phase 1 enzymes that increase the hydrophilicity of toxins, as well as a large amount of phase 2 enzymes that add a polar group to the toxin to facilitate excretion. Hepatocytes are involved in the synthesis of blood clotting factors, transporter proteins, cholesterol, and bile components. Furthermore, hepatocytes take up and store glucose, minerals, and vitamins from portal and systemic blood. There are various mechanisms of toxicity of nicotine. Firstly, nicotine competes with acetylcholine for binding to specific membrane receptors, nicotinic acetylcholine receptors, or NACHRs, which are widely expressed in various types of cells. When nicotine binds to the NACHRs, the conformation of the receptor changes, causing the channel gate to open up. This causes an influx of sodium and calcium ions into the cell, which results in membrane depolarization. This activates further influx of calcium ions and triggers a cascade of signal-dependent activity. As can be seen in the accompanying diagram, nicotine induces a growth pathway consisting of growth factors RAS, RAF, MEK, and MAPK, as demonstrated by the largest red arrow. The activation of this pathway results in the inhibition of apoptosis and increased proliferation. Nicotine causes the survival of cancer cells via the phosphorylation of AKT. AKT, also known as protein kinase B, in short, is a central player in the regulation of metabolism, cell survival, motility, transcription, and cell cycle progression. In the specific case of nicotine-induced calcium ion increase, the phosphorylation of AKT leads to the blockage of apoptosis and increased proliferation. The activation of the growth factor pathway previously mentioned that included RAS, RAF, MEK, and MAPK, as well as AKT, leads to the activation of downstream transcription factors AP1 and VDR to bind the promoter region of alpha-9 NACR. This results in a strong positive feedback loop, which ultimately leads to tumor progression. In short, an increase in calcium ions leads to the promotion of cyclin D1 transcription and translation, which increases mitosis. It also inhibits P21, a downstream product of P53, as well as other cell cycle arrest factors. It causes the activation of MYC, which leads to the activation of cyclin-dependent kinases. Cyclin-dependent kinases, or CDKs, cause the release of E2F, which transcribes genes necessary for cell cycle progression and mitosis, and activates E2F itself. Therefore, the activation of CDKs results in increased mitosis activity. The second mechanism of toxicity is a result of the pearl cycle opening. The amine function of nicotine may react with nitrogen monoxide or nitrous acid, which produces a nitrosonium-type molecule, which may then be oxidized and opened. Opening leads to two potential isomers, or two nitrosamino-type molecules with the formula R2N bonded to another nitrogen that is also double bonded to oxygen, where one of the R groups is a methyl. In an acidic environment, the oxygen of the nitrosamino group can react with another amine, which removes the positive charge from the nitrogen. If this other amine is a part of DNA, an irreversible DNA adduct has been formed. This alkylation helps in the development of cancer as it prevents the normal development of the cell. Nicotine is both a stimulant and a relaxant, which leads to mood-altering effects. These effects include relaxation, sharpness, calmness, and alertness. The stimulation effects are caused by the release of glucose from the liver and epinephrine from the adrenal medulla. The depression is caused by cotinine, a prominent nicotine metabolite. Nicotine is responsible for the subjective pleasure of smoking and induces the addiction. Nicotine-rich blood reaches the brain and causes it to release various neurotransmitters and hormones, including acetylcholine, which enhances concentration and memory, norepinephrine, which increases arousal, and beta-endorphin, which reduces pain and anxiety. Overall, nicotine improves cognitive function, including learning, memory, and attention. Nicotine is mainly associated with tobacco products, which result in an increased risk of cardiovascular disease and cancer. However, it is unknown which specific compounds cause those effects. Nicotine can potentially be used as a therapeutic to combat ulcerative colitis, or UC. Studies show that there are developmental and protective effects on UC from nicotine. The disease is less prevalent in smokers over ex-smokers over non-smokers. The protective effects are due to the nicotine and the carbon monoxide when smoking, but the precise mechanism is still unknown. Another disease that may benefit from nicotine is Parkinson's, a neurodegenerative disorder which impairs motor activities. Those suffering from Parkinson's have a decline in nicotinic acetylcholine receptors, NACHRs, and thus affect the nicotinic cholinergic system. Nicotine may have neuroprotective effects. Nicotine administration increases dopamine release from the striatum, which stimulates NACHRs on presynaptic dopaminergic neurons. Nicotine treatment reduces the behavioral defects and lesion-induced loss of striatal dopamine transfer.